Okay, hi everybody. Good morning, and thank you for joining the day two of the INC annual meeting. We are going to get started momentarily in just a few seconds. So, before we go forward, I just wanted to thank you again for joining this meeting today, and thank you for joining the meeting yesterday too. I hope you all had a great day, enjoying the gene therapies and cell and gene therapies and discussion on ethics yesterday. And the topic of the discussion today for our annual meeting is real world data. I'm going to go through the session with you all in a little bit more detail right after this slide. But first, I wanted to remind you a couple of housekeeping items. First one is that all of this today's session as well as the entire meeting is being recorded, and the videos of this recording are going to be posted after the meeting at the CPATH INC website. This website currently also holds all of the on-demand resources, including speaker bios, including detailed agenda of the session. So I encourage you to go to this website and take a look at everything that's been happening in this meeting so far. I really do not think that it's actually going to happen, but just in an off chance there are any technical issues or any serious WebEx platform outages, we do have a backup plan. We will email you with instructions. So if, let's say, WebEx suddenly dies on you, on everybody, we will send you an email. So do take a look at that email and look for it, which I really do not think it's going to happen though. Next is, in case we need to upload any pre-recorded videos for broadcasting, the audio for those videos is only available if you are using your computer audio to listen in, and if you're calling in through phone, then you would not be able to hear any videos at all. So as a best practice, please use your computer audio and do not call in using your phone. Then we would obviously appreciate if you would share our event and share the news of our event on your social media using INC 2021 hashtag. Another very important point is that do not use VPN connection to connect to the WebEx, because VPN can certainly cause audio issues, and it can cause issues with connecting to the WebEx in the first place. So as a best practice, do not use VPN when you're connected to the session today. Uh, we encourage feedback to everything that we are doing today, and we encourage the attendees to ask questions. So if you have any questions for any of the panelists, for anything at all, please use the QA panel on the bottom right of your screen and enter your questions and direct them to all panelists. So it's important, because otherwise, if you direct it to some specific panelist, nobody else in the panel would be able to see those questions. And the purpose of this QA session is to encourage a natural and mimic a natural conversation amongst a group of people. So those were some of my housekeeping items for you. I'm just gonna go jump into the agenda of the meeting today. So as I said before, the topic of this discussion is today is real world data. So real world data is basically data that you collect, that is collected in a routine fashion, in a real world fashion. If many of you in the audience might be neonatologists, and then if so, you would know that neonates are basically called as therapeutic orphans, in the sense that over the years there have been very few drugs that have been specifically developed and targeted for the neonatal population. And that hampers basically everybody's ability to use some of that data to generate further evidence. Fortunately, neonatology is also a very data-rich specialty. There's tons and tons of real-world data available, and the purpose of this session today is to discuss how that data that is already available can actually be used to advance and accelerate drug development in the neonatal population. So we are going to start with introduction by the chairs of the session, that are Klaus Romero, who is the Chief Science Officer at Critical Path Institute, and Nina Modi, who's a Professor of Neonatal Medicine at Imperial College London. And then after this introduction, there's going to be a couple of keynote speeches. First one would be a parent-patient perspective, voice of the parent, speech by Deb Dicenza, who is the founder and CEO of PremiWord and is also on the leadership team of International Neonatal Consortium. Then there's going to be a keynote speech by our FDA colleague, John Concato, who's the Associate Director for Real World Evidence within Office of Medical Policy. policy. Subsequent to that, there would be a speech on presentation on considerations for real-world data in neonatology by Ralph Bax, head of pediatric medicines at European Medicines Agency. Then there's going to be a presentation by Critical Path Institute, Data Collaboration Center, and Quantitative Medicine Division within CPATH. Following that, there's going to be a panel discussion and a QA session, for which we have several panelists who are going to present diverse points of view depending upon where they're coming from. We have Satoshi Kusuda from Japan, Mike Padula from Children's Hospital of Philadelphia, Laura Fabri from Chiesi Pharmaceuticals in Italy, Andrew Brammer from NICHD, Colin Hovinger from Iact, Shirley Porkes from she's the faculty of pharmaceutical medicine and the director of Actors Consultancy. And then at the very end, we are going to have an award ceremony, Data Pioneer Awards, and after that, we will close for the day. And now, without any further ado, I'm going to invite the chairs of the session today, that is Klaus Romero and Nina Modi, to provide introductory remarks for the session today. So first up, I'm going to ask Klaus Romero, who I said already he's the Chief Science Officer at Critical Path Institute, and he'll come in and provide you with brief introductory remarks. Klaus?
Excellent. Thank you, Ken. Would you just a quick check that you can hear me okay?
Yes.
Fantastic. Okay, so yeah, thank you everybody for being here. This is a very exciting time, and this is a very exciting meeting for us at CPATH in general, and in particular for the International Neonatal Consortium. The commitment and the support that we have received from the FDA over the years is patent and even stronger for Inc. And as such, CPATH and Inc. This is your consortium, and we want to reiterate our commitment as a neutral third party to ensure that we give it back to the agency and to industry, but ultimately to the patients and their families. That we work on leveraging our core competencies to transform patient-level data into actionable solutions that can accelerate medical product development for this population in need. And one thing that I find really exciting is the fact that in the past there's always this notion that neonates come last, and so there's adult development, and then there's some pediatric extrapolation, and then some further extrapolation to the neonatal field. With the work that we're doing in real-world data and transforming this into real-world evidence, with a focus on making that evidence truly actionable to accelerate drug development, the neonate community is going to not only benefit, but also generate some really powerful learnings for other areas. So I'm really looking forward to this transformation of the neonatology field, teaching the other fields some pretty valuable lessons. So that's another area of great excitement for us. And so we're going to cover today the aspects of that work. And so with that, I'll turn over to my partner in crime, Nina. Take it away.
Thank you very much, Klaus, and hello everyone. It's great to be here. I think I've been involved with INC since it began many, many years ago. We had a first meeting in London, and it's been absolutely fantastic to see Inc. go from strength to strength and for its community to widen and enlarge around the world. And I'd also want to echo Klaus's comments because, of course, as a neonatologist, so I'm a clinician scientist, I'm a neonatologist, but I'm also a researcher and the head of a research group, and also have a few professional leadership roles. But I would echo Klaus's words, and that is that it's high time that we broke down that old myth that children and babies and mothers come last. That they absolutely come first. They come first in the biological cycle, and they should be orphans no longer. So my interest with Inc. is not only because I'm a researcher and because I'm a neonatologist and I'm a mum, but I'm also, of course, the head of, I've led the development with a lot of colleagues of the UK's National Neonatal Research Database, and am now leading, again with lots of colleagues, an international neonatal research database. And we are the living proof, I think, for the UK that we can take real world data from babies' electronic patient record systems with the support of their parents and colleagues various, transform, curate that data, turn it into real world data that is high quality, that can be used for a range of diverse applications, extending through from audit, quality improvement, surveillance, health services research, clinical research, real-world clinical trials, artificial intelligence applications. The data is the data is the data. And I think we have a responsibility to use the data for multiple purposes which benefit patients first and foremost. So I'd like to say congratulations to Inc., all my colleagues there. You've done fantastic work over the years. It's amazing how the years go by, but they do go by when we're having fun. And I think that apart from, breaking down a few barriers, we have had fun together, and I hope we're going to see even better improvements through real world data for the patients who we are here to serve. So many thanks, everyone, and I look forward with great interest to listening to this afternoon's session. And my final word will be to say special thanks to Bayer, Yazi, and our UK National Institutes of Health Research, because without their help, we would not have got our new international neonatal research database off the ground. So special thanks to them.
Thank you so much, Nina, and thanks, class. And now we're gonna go directly into the sessions for today. But before we start, it's as Nina said, that everything that we are doing, and then as Klaus also said, everything that we're doing here is for the benefit of the patients. And that is why what we really wanted to do today was to start the proceedings by starting with a keynote from our parent representative, who's Deb Dysenza. She's going to provide her perspective as a parent, and also as a patient, because she was a preemie when she was born, and she's going to talk about why these type of data sharing activities that we're going to be talking about today are so vitally important and so critically important. So I'm going to invite Deb Dysenza, who's the founder and CEO of PremieWord, and also she's also in a leadership role at International Neonatal Consortium, to provide a few words and few remarks.
Thank you, Kanwaljit. Thank you, everyone. It's an honor to be here. It's an honor to be a part of Inc. and to do what we're doing every week, every day that we meet. This is vitally important to me, obviously as a parent, and yes, someone who was born prematurely as well. You'll hear about that later. My daughter is a really good example, for a variety of reasons, why real world data is necessary. She was my 30-weeker, so she was 10 weeks early, and she was the 2-pound, 15.5-ounce baby. And we heard lots of things like she'll be fine, she's doing great, everything's great, and we took great heart in that. Fast forward, she spent 38 days in the NICU, and it was anything but fine. It was very much the roller coaster that parents talk about. We almost lost her several times, and she almost had to have heart surgery. We were told the cranial ultrasound was clear. Turns out it wasn't. Just a lot of problems, a lot of issues, a lot of breathing problems. And she came home on oxygen and a monitor, had a lot of feeding issues at home, and went back in the hospital, which I gather is common. And she came home on full-time oxygen. I have to say that it was at this point that I was starting to really question people and question things. Whereas in the NICU directly, I just believed everybody around me was the expert of my child, and that I wasn't. Very quickly, I was getting to know my daughter outside the womb. And I feel like that was the moment that I started to gain my feet as an advocate. And that was so important, because I started speaking up when she had the readmission. I started speaking up for a variety of reasons in the NICU itself, but I just really didn't gain my feet with that until after the NICU. And I was that parent in the NICU that was asking questions basically around development. Was she going to be okay? Was she going to walk? Was she going to talk? But I didn't voice that because, again, I was the impotent parent. I was thinking this the entire time. Is she going to be okay? Is she going to be able to ride a bike? I didn't know. Well, she came home. She didn't qualify for early intervention. And she said her muscle tone was great. It was wonderful. Okay, good. Fast forward to 18 months, we had the local health department following her. It was an option on the discharge paperwork. They followed her and then told us at 18 months of age, she needs to get checked out by early intervention. She probably won't qualify, but please go get her checked. Okay. She got in so fast, my head spun. I have to say, she got in and they were all over her, making sure she got help with physical therapy, occupational therapy, speech therapy. And I knew there were some things wrong, but I think I was also in denial because, again, we were told she's fine, she's doing great, she's fine, she's fine. I get that she lived. I get that. But fine means many different things. And so fast forward again, she got out of early intervention. She went into preschool, and I mentioned to her preschool teacher she was premature. I was very upfront about that. I wanted to be proactive and not reactive. And the preschool teacher kind of went, oh, yeah, but they catch up by age 2. And I'm like, she was a preemie. I want to be proactive, not reactive. That preschool teacher heard me. And at the first parent teacher conference, as my husband and I sat there, and we're excited. We're having this parent moment. It felt normal. It wasn't normal because from behind me, I heard the teacher come in, and as she walked toward us, shut the door and walked toward us, she said, well, you obviously know more about this than I do. Becky wasn't speaking in school, and she urged us to go push for child find services. And so we did. That journey led us to kindergarten. Kindergarten around an entire room of people, 10 people around the conference table in the principal's office. Becky, they started talking to me about how she was doing, and I was the one speaking up and saying, look, this is a problem. Something's wrong. I'm not sure what's going on. Well, Becky ended up getting diagnosed with autism later that year. I was in shock. By second grade, I told her pediatrician, I told him, I said, we are talking about ADHD. Is this a comorbidity of autism? He said, yes, it's very common. He sort of ignored her autistic diagnosis, by the way. He acted like it wasn't a big deal. And then the ADHD diagnosis happened, and he was like, okay, it's fine, come back for medication, et cetera, et cetera. All this time for probably since she was diagnosed or she was in early intervention, I've been wondering about her walk, her gait, her running. I've been asking professionals, a developmental pediatrician, ultimately an orthopedic surgeon when she was having squishy ankles on an elliptical. All this time, we ended up with a CP diagnosis when she was 13 and a half years old. She had just had her bat mitzvah 7 months before. I feel that all the data in the world failed us in this experience. I feel that Becky did not benefit from the small studies that are out there. As far as the medications go, she benefited from what was there, but it's not enough. Today, she is 18 years old. Today, she's a smart cookie, and she stands up for herself because we've taught her so. But I have to say, I look back at her whole journey and I look at everything that data could have done to help her, and I don't feel we got what we needed. And I feel that we are on the brink of something massive here with INC and all that it's doing with this real world data project. I feel that we are on the brink of making massive efforts towards new medications, things that can improve outcomes, like children like Becky. It can improve outcomes in so many ways so that I don't have a 35-year-old who is currently getting diagnosed with cerebral palsy. She's 35 years old. I'm walking her through it. And I'm just stunned. She's one of John Davis's surfactant babies, as it turns out. And I sit there and I think about this and go, wow, what the data could have done. It could have made not only help babies survive, but truly thrive and not struggle in school, and not struggle on the soccer field or the football field. And I worry about that. I worry about these patients because I'm working alongside them and with them, and they're coming to me and telling me their stories. We've got to do better. And we cannot work in silos doing that. We cannot be small sample sizes and studies. We have to do better. And that's when we work together to do it. And that's what so many people that you will hear about today getting their awards, what they have provided. It is totally possible to do this. So I urge you, join in. I am working on a project called Preemie Crystal Ball, which is a data portal around long-term outcomes of premature infants. I have to say that my hope is to insert my data into INC, to share it for the benefit of all. Because as everyone's saying, it's about the patients and the patient family. We need to do better, folks. Work with me. Talk to me. Tell me why you are hesitant about sharing the data. I think you'll find that it's okay to do. It's important to do, and it's the right thing to do. Thank you.
Thank you so much, Deb. That was really so poignant and, on the mark. And that is exactly why, what we are doing here is encouraging people to collaborate with each other and rather than being in silos with their own silos. That is why, whatever we are doing is so important. And that is why your voice as a parent is so critically important. So, thank you so very much. We greatly appreciate your thoughts. And I hope you'll stay with us in this panel discussion today. And at the end, I'm sure there are going to be a lot of questions from the audience, and we'll invite you to provide your feedback further at that point. But again, thank you from the bottom of my heart for your comments today. Thank you.
Thank you.
And then next up, we have our keynote speech coming up by Dr. John Concato, who is the Associate Director of Real-World Evidence Analytics within the Office of Medical Policy at CDER, FDA. And Dr. John Concato is going to talk about real-world data, real-world evidence, and public health. So Dr. Concato, I'm going to give you the presenter controls and the mic, and you should be good to go.
Thank you, and thanks to Inc., to CPATH, and to Deb for her presentation. I'll follow with a high-level overview of FDA, real-world evidence, and public health. And this first of two background slides is actually aligned at a very high level with what Deb just presented, much less detail and much less personal implications, of course, but we should recognize that the types of healthcare range from health promotion to disease prevention to diagnosis and treatment to rehabilitation and to palliation. And I'm not bringing in bedside manner and patient level data just yet. But that applies, of course, across the age span, across the lifespan, starting with neonates, which are an under-studied group, infants, child, adolescents, and of course the adults. Too much to fit on one slide. But I just wanted to start again with a very high background landscape slide. And to that I'll add, we are currently in the era of big data. Actually, the term big data appeared in the computer science literature first, apparently, during the 1990s, often referring to data that were too large to be stored in then conventional storage systems. But well, a not so recent article from about 7, 8 years ago said it's unclear when big data became the buzzword of the day or really what it means. And I think to a certain extent that's still true. But one perspective is that modern technology has increased the quantity and forms of available data. We don't go back to paper charts very often anymore, as well as the speed to merge and manipulate data. Yet we also should remember that integration and analysis of large-scale data has always been integral to epidemiology. So there's some new aspects, but there's also some aspects that are tried and true. Let's now turn our attention to the FDA as an agency and the Center for Drug Evaluation and Research as a center in that agency. The FDA's mission statement is that we're responsible for protecting the public health by ensuring the safety, efficacy, and security of human and veterinary drugs, biologic products, medical devices. I won't read the whole slide. I will say that I'm representing CDER and to a lesser extent CBER, where CDER's mission is to protect and promote public health by helping to ensure that drugs are safe and effective for their intended use. They meet quality standards and they are available to patients. In this case, we're focusing on neonatal patients. It might be said that that big data in healthcare arrived at the FDA officially with the 21st Century Cures Act in 2016. Most of us know this, so I'll be very brief, but FDA was mandated to establish a program to evaluate the potential use of real-world evidence to either support a new indication for a drug already approved or to satisfy post-approval study requirements. We were to issue a draft framework by December 2018 and draft guidance by December 2021, while we maintained our thresholds for substantial evidence. And we can also say that the Prescription Drug User Fee Act, PDUFA, commitments are aligned in this way. So nothing new here, but also not so new, but to point out that the RWE framework does apply to both the Center for Drug Evaluation and the Center for Biologics Evaluation. CDRH, to their credit, has their own program, their own guidance, their own framework. But I will just mention briefly that we have a multifaceted program intended to, again, improve the public health generally, including in the neonatal space. It includes internal agency processes. For example, just how the review divisions are equipped to review real-world data, real-world evidence, external stakeholder engagement. I guess today's session is related to that, as an example of that. And then I'll have just a couple of slides on guidance development and demonstration projects to give a flavor of what the FDA is doing in this space for those who are less familiar. But first, again, most everyone might have seen this, but I'll just be brief to say that the definitions of real-world data and real-world evidence from our framework include that data, real-world data, are data relating to patient health status or delivery of healthcare routinely collected from a variety of sources. We tend to think of electronic health records, EHRs, medical claims, registries. It's certainly patient-generated data, as we heard, is very important, and other sources, including wearables, which currently have some traction more in the adult space, of course, but you get the idea. And then real-world evidence is defined simply as evidence regarding the usage and potential benefits that are derived from real-world data. And importantly, that is not synonymous with observational studies, but rather randomized trials, pragmatic trials, for example, externally controlled trials, as well as observational studies, can use real-world data. And the definitions work well, but this next slide, the title actually introduces a bit of caution. Randomized, observational, interventional, and real world, what's in the name? I don't want to say more than a couple of words about this, but in the middle of that text there, it starts by saying FDA is evaluating how observational studies can evaluate efficacy. We mean that with sincere, but in this context, a randomized trial versus observational study dichotomy is overly simplistic. And I use it as shorthand in day-to-day work as well, but just let's be careful when we're submitting proposals to FDA or certainly submitting drug applications to be clear on what we mean, whether this is an interventional or non-interventional design, whether we're talking about primary data collection or secondary use of already collected data. And there's more issues, but it's just a plea for a little bit of clarity, because it tends to be too easy to just say real world evidence, real world data, and not be specific about what exactly we mean. And this next slide actually is sort of FDA's, or I should say CDER's, current landscape view, where we have, going from left to right, you see at the top row, randomized interventional study designs, non-randomized but still interventional study designs, and then finally the non-randomized, non-interventional. If this is a little jargony, please bear with me. But basically the bottom row shows that there's an increasing reliance on real world data going left to right. So we can have even a traditional randomized trial that uses elements of real world data in the upper left there. I mean, I guess we've always done this, but literally real world data can be used to better determine enrollment criteria and trial feasibility, for selected outcomes, to support site selection, or to capture supportive endpoints. As we move towards the middle, we have trials in clinical practice settings, often called pragmatic trials, where we could use the EHR, for example, or claims data to populate an electronic case report form, still in the randomized trial. It is true, most people think that to the right is what real world evidence is, what we're talking about, say a single arm study with an external control arm from a real world data source, or a standalone observational cohort study where all the patients receive the test article, the drug of interest, in clinical care. So that's as technical as I'll get, but I did want to include a bit of a detail just to orient us for this discussion. But here now I'm pivoting to an example of guidance development. This guidance was released in September. It's all about assessing electronic health records and medical claims to support regulatory decision-making. So we're very pleased to see this get through clearance. And I don't think I'm violating any policy to say, please stay tuned tomorrow or Friday for a second in the series of FDA guidances in the real-world data, real-world evidence umbrella that will come out. We're releasing them as soon as we can, as soon as they get through clearance. And they are intended to help stakeholders as well as academicians who aren't necessarily going to submit to FDA. It can't hurt to look to see what the agency's thinking is. I was asked to be brief, so in that I'll move on now to demonstration projects or development projects as another bin, another category of our multi-pronged approach. We have upwards of 20 demonstration projects, either completed, ongoing, or new. This is in the new bin. And to be brief, this was under a UL1 HHS Health and Human Services mechanism. We received 31 applications. They were all actually good, but we could only fund 4, sort of NIH payline style, unfortunately. But the 4 applications that were funded include a variety of applicants in that middle column there. We see Brigham and Women's Hospital, Genentech, Barantos as a private entity, and CPATH. The titles reflect the focus, which is also varied. In one case, the first row is enhancing evidence generation by linking randomized trials to real-world data. So that supports the left hand of my landscape slide, if you remember that image I showed. The second row is applying novel statistical approaches for so-called hybrid designs, where you combine an internal control with data from real-world data sources. So again, we want high-quality evidence, but it doesn't always have to be with the traditional randomized trial each and every time. The third row is transforming unstructured and structured data to advance tailored therapy. We know that EHRs have a lot of information, but we also know that unstructured data is harder to get at and hard to work with. That's a very simple overview on my part, but this particular project is one of many efforts in this field to be able to get at and work with unstructured data. And last but not least, and I didn't know I'd be here today when there was an internal peer review process involved that I was part of, but not a reviewer proper, but advancing standards and methodologies to generate RWE from RWD through a neonatal pilot project. So front and center for the discussion today. And well, all I did was go back to the proposal and quote from what was submitted and was eventually approved and awarded. Each year in the US, 10% of neonates are born preterm, and there is an urgent need to improve survival and outcome. However, there is minimal new drug development, and most existing drugs have insufficient evidence to support safety, efficacy, and dosage in this high-risk population. The grant application went on to say this project will greatly accelerate neonatal drug development by collecting, extracting, curating, standardizing, and analyzing RWD to generate RWE and addressing major challenges in neonatal drug development. I'll just leave it at the project is off to a great start. There'll be more discussion of it, but I'm going to leave it there in the interest of my overview and move on actually to briefly talk about outside of the neonatal space, what has FDA done? Well, actually, this slide shows that even before the 21st Century Cures Act or the era of 21st Century Cures, what we now call real-world evidence was being approved under the existing regs. So that's good news in that we don't need to sort of reinvent or invent a new wheel. I won't go through the details here. I'll just say, look at the third row, blinatumomab for acute lymphoblastic leukemia, excuse me, I'm rushing. Single-arm trial with a reference group that consisted of patient-level data on chart review of almost 700 patients in Europe and the US. So this we would now call a real-world data study using EHR data. But again, this is pre-21st Century Cures. The next slide is in the 21st Century Cures era. And again, you go from left to right. It's the drug, it's the indication, when it was approved, and where the real-world data came from. Here, if I'm going too fast, please bear with me, but I'll just pick row 3 again. The Zostavax example, it gives me a chance to give a nod to our colleagues in CBER, the Center for Biologics, where they used a prospective observational cohort study, again, happens to use EHR data, to characterize the duration of protection in persons 50 years of age or older, so clearly not the neonatal space and also not the basis of approval, but it's an example of how real-world data, real-world evidence can inform effectiveness across the broad spectrum. This next slide is probably the latest installment of that, and I do want to spend just a little bit of time on it. FDA approved the use of a transplant drug based on real-world evidence. So this is Prograf tacrolimus for the prophylaxis of organ rejection in patients receiving liver in 94, later for kidney and heart, and the drug is widely used in clinical care. But while RCTs, RCTs were done for lung transplant, but not submitted to FDA, I should really tweak that slide. In that context, Astellas, as the sponsor, submitted a supplemental new drug application to FDA with what we would call a non-interventional, quote-unquote, real-world evidence study. The study data and design were evaluated, maintaining our standards, and we approved the drug for preventing rejection and death in lung transplant. Now, why might this be considered more than just another approval? What's not on the slide is that the non-interventional, aka observational study, was considered the adequate and well-controlled investigation. So for those who are familiar with US regulations and statute, this is a quote-unquote big deal. This is the first time that so-called real-world evidence was viewed as being adequate and well-controlled, and actually the trials in other settings, liver, kidney, heart, were viewed as the confirmatory evidence in terms of the structure of how we approve drugs. So it shows that the FDA is honoring its mandate, and this is only one drug. We're not resting on this, but we will move forward with lessons learned from this, which are all positive, I would say. Next, I want to just, everything I've said gives us reason for hope, but I think it's also important to show that we do have to pay attention to what has been called hype. The x-axis here is time, the y-axis is expectations. Many will have seen this. The innovation trigger at the origin is basically the era of big data, which I alluded to earlier. This shows a peak of inflated expectations, a trough of disillusionment, a slope of enlightenment, and a plateau of productivity. It's not as if, well, maybe we could put all of real-world data, real-world evidence on this, but I think it's more whether it's data, design, different aspects of real-world evidence are at different points, and actually any one application or submission to FDA might fit anywhere on this curve. But I basically just want to say that we're looking forward to working with everyone, but having real-world data does not automatically generate real-world evidence. Yet efforts such as today's program will help us get to that plateau of productivity, which is where we want to be. I believe the next slide is my wrap up, my summary. Again, since I'm the FDA representative here, speaking for myself and not for the entire agency, but I think it's fair to say that as a summary that we did establish a program to evaluate the potential use of real world evidence. Our framework has been released for almost 3 years now. It's been very well received and referred to often. We have published and will continue to publish draft guidance, which will become final guidance in the next year or two. And at the same time, we, on behalf of the patients who are here, nothing worse than the FDA approving things that turn out to be not worthy of approval. So we're balancing that with also not delaying useful medications to the bedside, but we will maintain our standards with substantial evidence. And we are dedicated to honoring our commitments to PDUFA, which is now going to become PDUFA 7 in short order. So my last slide is just an email address if anyone wants to reach out, either on the basis of this presentation or in other contexts. Thank you very much for the time, and I'll pass the baton back. Thank you.
Thank you so much, Dr. Concato. That was an excellent presentation, and I do see that there are already questions coming in for you. I'm going to save them on until our panel session today. So I'm just going to go directly on to the next slide. If you could just advance to the next slide, please. Okay, thank you. So next up on our list of presenters is Dr. Ralph Bax, who's the head of pediatric medicine scientific evidence generation within European Medicines Agency. We are so honored and lucky that Dr. Bax agreed to make a presentation on real-world data today. We have been, INC has been working with Ralph Bax and EMA ever since day one on a variety of different activities. So Dr. Bax, I'm so glad that you're here today and that you're going to talk about considerations for real-world data, real-world evidence in neonatology. And if I'm going to pass the slides back on to you today.
Okay, thank you to everybody, and thank you also for your support over the years in the area of neonatology. And as you know, we have had the pediatric legislation in Europe for over a decade now, and when this outcome, or when this work was analyzed, unfortunately still areas like neonatology and pediatric oncology need further support, need to be pushed further. So any activity in this area is really needed. And we also did have changes in the way that across the pediatric investigation plans, it was tried that neonates don't come last, but this change takes a while, and of course regulators, but also trialists and industry, of course there is some risk aversion, and therefore we are also looking with great hopes to new endeavors and new ways of shaping. No, I'm pressing now the Zoom instead of, wait a minute, sorry. All of this needs practice, but now, yeah, this is my disclaimer that I'm speaking, I am expressing my personal opinion here. And indeed, we need to advance in how we generate clinical evidence. And here I'm not speaking, as you have seen, as a specialist in real world data or real world evidence. I'm rather speaking as a part of a learning organization and a future user of these opportunities and possibilities. So we are seeing the drivers, and again, I would like to reflect here a little bit mainly that actually data and our analytical capability and the capacity, these are all enablers, and they should actually contribute to what we are trying to achieve and to further develop. And here I would like to, in my short intervention, I would just like to reflect a little bit on the different perspectives of data being used, real world data being used by regulators themselves and provided, for example, for submissions for marketing authorizations. And here I'd really try to be rather operational, taking more the perspective of how the PDSO, so the Pediatric Committee, might use these opportunities and is currently trying to learn and use this in several projects. And here I just wanted to emphasize again that transparency, reproducibility, and replicability of results is really very important. And but that has already been mentioned, and experts know these criteria anyway. And this is also a little bit repeated here. I would like to add on this slide is the timeliness. So what the agency is planning is to have a sort of service that actually also regulators and committees can use these new methodologies also themselves to complement what an applicant has provided. And for sure, therefore, we need to be able also to have fast access to data and to these methodologies. Here you can see what I just said. So on the one hand, it's use to support PIPs in our case, and use of these methodologies by the regulator versus looking at the data provided by pharmaceutical companies. And finally, this should then support, in our case, of our own use, and also obviously of the real-world evidence provided in applications, finally to support the committee decision-making. Traditionally, it works obviously, as you know, in a way that applicants and drug developers provide the evidence, they perform the studies, and the agencies wait for the results and then to evaluate. This is now also changing, and as I have mentioned, by becoming more and more proactive and getting also support for our own assessment using these methodologies, here we are really putting great hope into RWE. And that is, at the moment, as I said, we are a learning organization. We have done studies using electronic health databases which are accessible, studies procured, and then finally there will be a development called Darwin. I just wanted to very shortly enter into this and then move on what we practically could be looking at as a first step. Here it's just a short scheme of how to improve actually the use and to improve the use of these new methodologies and analytics to use it for decision making. So it is obviously the supply and also demand. And in our case, on the demand side are the scientific committees, the various national competent agencies, and all other bodies involved in drug development. And as has been said, we have to develop skills, we have to make data available. And here I would also like to emphasize what has been said yesterday, the importance of the engagement and activities like CPAS is really not replaceable, and we have to rely on it as academia by themselves and even industry will not manage without such support. Here, just coming back, there have been studies on electronic health databases accessible in-house. So there are 3 databases where we have access, and the main focus obviously was in the area of safety. So here we are already using it regularly, and then further, similar to FDA, studies have been procured through, in this case, EMA framework contracts, and have been here I have given below. You can see some examples in the area of COVID, but also, for example, potential risk of cancer associated with NDMA. And these studies are being extended. And finally, a federated network is planned, as most of you will know, and this is just to mainly bring together all the partners needed in the European landscape in order to provide expertise and also services. The regulatory use is the uses, as colleagues have expressed, are numerous, and in order to a little bit show the complicated European structure, here is a scheme which goes through the so-called life cycle of the drug development, pre-authorization, evaluation, and post-authorization. So in the pre-authorization space, you have right in the middle, for example, focusing here on pediatric investigation plans, the PDCO, and then it moves on finally to evaluation and post-authorization, different areas where all this can be used. Finally, obviously, the need is also here to get support by funding and by providing opportunities to qualify for qualification of methodologies. What we do want to avoid is the so-called black box. We have to convince users on all sides on the criteria and of validity as a transparency, as I have explained, to reach a level similar to clinical trials where you have this structured approach. And we believe this just will support also its use, but at the same time, as John has mentioned, put it exactly there where it is needed and where it really can also contribute. Data in itself are just as such, as you know, are not sufficient. And from our perspective, so from a regulatory perspective, it has been mentioned. I just wanted to go into a little bit more detail. So focusing on the first two columns, the use case is, on the one hand, supports the planning and validity of applicant studies and understand the clinical context. I will focus on these. Of course, also use is seen in investigating associations and impact. And here, as mentioned also, we are planning to get support in the area of design and feasibility of planned studies, which are submitted to us, representativeness and validity of completed studies, to understand the clinical context in view of disease epidemiology and clinical management and drug utilization. As you can imagine, neonatology is a prime area for these requirements, because here we really often struggle in finding the exact information in order to support the optimal pediatric investigation plan, so the plan for the development of the drug. And as you might know, we are here really at an early stage of development. Applicants are asked ahead of their program to agree this plan with the agency, and the FDA has obviously a similar program. And in this early stage, such tools would be very valuable. So informing, as you can see here, about recruitment, examination of impact of planned inclusion, exclusion criteria. So all these, so even to evaluate traditional randomized clinical trials, this additional tool is seen as very valuable, and we are practically starting with these steps. And that we heard yesterday about biomarkers. I don't have to go further into this, endpoint validation, and then providing information on decisions to optimally select patient baseline characteristics. And yeah, you can read the rest. That is supplied more as information. And also in neonatology, where we really struggle is the number of patients, the incidence and prevalence. It sounds rather a banality, but it is amazing what we still do not know. And as one of our tasks is also to support applicants, to support drug developers, yes, also to push them. It is such a database, and such data are of real value. It goes on. We can't rely anymore only on published literature. If we can, also in a timely fashion, get really up-to-date information, this would help, even if we are still at a rather traditional stage of development programs. It would help in even advancing those. Here, I just wanted to show you what has been planned concretely in the area of the Pediatric Committee. As you know, we are having pediatric investigation plans, which also can be waived. So here we have a lot of discussions with drug developers on which direction we should go. And one of the points is indeed disease epidemiology. We do not want to push for a development of a drug which finally is not needed. And that was obviously the danger also in the past. There was a drive. We didn't have, or still don't have, sufficient neonatal drugs. However, we should push really in the right direction. For PIPs, it will be the assessment, as I said, on design and feasibility of planned studies, the representativeness and validity of completed studies, disease epidemiology, and clinical management, as a PIP discusses the complete development from quality, non-clinical, and clinical studies. All of this is seen as a supportive measure at the moment. And of course, as a regulator and a learning organization, it also means that we need to learn how to assess real-world evidence and potentially use ourselves real-world data for an in-house analysis. So overall, this is an exciting development, and I would like to conclude that we need a stepwise approach to allow the system to evolve and understand how best to use these concepts together with the current concepts. And here we would like to emphasize the need for validation and qualification procedures could be used, for example, in this matter. Then bear in mind the different perspectives of real-world evidence generations by regulatory bodies themselves, as I have tried to explain a little bit, and by drug developers. Discussing with PDCO in view also of this session, colleagues emphasized practical hurdles. So we might be in Europe not so far yet. When I hear the advances in other areas of the world, so colleagues still cite practical hurdles and including obviously dealing with European data laws, but everybody is looking forward, specifically in neonatology, for example, in the area of natural history data and the support of design of clinical trials. I mentioned the ongoing pilots, and I hope next time we will be at an advanced stage and can report much more in this area. Thank you very much for your patience and for this meeting. Thank you.
Thank you so much, Ralph. That was great indeed. I do see that in response to your presentation, several questions have come in, and I will get to them during the panel session and hope you'll be able to stay on to answer some of the questions from the audience. But I will move straight on ahead to the presentation by our Critical Path Institute team. And first up, we have Will Roddy, who's the Senior Data Engineer and Team Lead at a Data Collaboration Center. He's going to talk about plugging in data, harmonization, and integration of real-world data. So, Will, I'm just going to pass the slides back on to you. And welcome.
Great, thank you, Kamalji. So I'm Will Roddy. Like Kamalji mentioned, I'm a data engineer on the data science team here at the Data Collaboration Center. I work closely with our data managers, our data scientists, and we work sort of upstream of being able to hand the data off to quantitative medicine. I'm going to be talking about some of the components of real-world data and evidence that we handle here at CPATH. I think that so far this morning and yesterday, we did a pretty good job of covering what real-world data is and where we're trying to get to that real-world evidence. But I want to call out some of the specific challenges, especially with the actual data. And so some of them that I'm going to try to address today that the Data Collaboration Center spends a lot of time thinking about is identifying the sources of data. And this sounds sort of obvious. You know, we say, oh, well, we want registry data or EHR data, but actually being able to get at that data and understanding, you know, who is the individual who sort of has the keys to the kingdom, so to speak, and can actually go extract the data is easier said than done sometimes, but also understanding where does that data live. If you need lab values, where are the lab values stored? What, you know, kind of where is the actual source that we're able to extract that data from? Tied into that is what is the security and patient privacy considerations? And this is something that's like of utmost importance to us. So we want to make sure that we're respecting patient privacy and that we're handling the data with proper security measures. So we're abiding and exceeding all regulations when possible. And then some of the other things is there's lots of harmonized data structures and there's varying data quality across each of these sources. I'll dig into those a little bit more. So just kind of a high level overview. What does it really take to go from a source to some kind of evidence? So we may have inputs such as natural history data or EHR data that may come from an EPIC or Cerner implementation. It may be coming from a clinical trial or from some kind of registry. The Data Collaboration Center handles a number of steps in the middle there. Things like we put it into a data vault, we do some curation, we do metadata annotation and standardization, all of this so we can surface the data through a user-friendly, secure cloud environment where other researchers and internally our quantitative medicine team are able to access that to create actionable insights and develop evidence. And so just to kind of give a high-level overview, I'm going to dig into each of these in the next few minutes. But data storage is, like I mentioned, important. We spend time thinking about how to secure and transfer that data. We apply anonymization techniques to ensure that privacy before we share it externally. We do some curation and harmonization, and then we transform that to a common data model. In the case of real-world data, we typically use the OMOP common data model or the Odyssey Communities implementation. I think one thing that can be a little bit confusing is what exactly is data harmonization? You know, what are we really talking about? And I think that a good kind of analogy is that you can think of data from multiple sources as like a big mess of tangled cables, where you would have cables for power or video or audio or other functionalities, and you're not even really sure what it does, but you know that if you unplug it, nothing works. And you end up with all of these multiple different kind of plug types that you can't really plug them into any two sockets, and they all look different. And nothing really fits in the right place. And so you sort of think of every data source as its own plug type, where registries are one plug and EHRs are another plug. And they're all transferring different information, but that we need all of it. And unfortunately, sometimes even data sources may have multiple plugs within one instance. So it can become a pretty complicated tangled mess. What we do at the DCC is we curate and harmonize the data where we transform these plugs into a single standard plug type. So you can think about it as some of those older cell phone chargers where you remember you could get the ones that would all convert it into a USB so you could charge it from one source. You can think of data standardization sort of along those lines, where maybe we have ICD-10 and ICD-9 codes and a clinical trial and a registry, and we hook them all up so we're able to use a common data model or just a USB. One thing that is difficult about this is that there's lots of different data paradigms. And sometimes we get lucky where there's already existing adapters. And you can think about those as like those iPhone Lightning to headphone adapters. So you are able to get, and I think they may even come with some iPhones now. And that's great. When we have those, that's awesome. We're able to just plug and play. But other times we need to do manual mapping where our data managers and our data engineers get into the nitty gritty and we're actually wiring up the connections between things and saying, oh, I'm gonna take this plug and put it here and I'm gonna split this one, and we're slicing and dicing the data to be able to make it work together. And all of this is to get it into the same format. But one of the challenges is that data comes at different granularity, and the granularity and the broadness of the data also has implications for what we can do with that. So once we've harmonized it into a single standard, we also apply hierarchical concepts. And in this case, I mentioned we use the OMOP common data model. We use their standardized vocabularies, but leverage several other source standards. So we're not inventing something new here. We use something else that's adopted already within the real-world data community. But a good example of where hierarchies are important for modeling data is thinking about drug types. So you can have a drug class, which would be like an NSAID, but you could have a prescription, which may be Motrin or Aleve, or Motrin oral suspension, 20 milligrams, or Aleve 220 milligrams oral tablet. And you can begin to see where this gets really complicated, that depending on the data source, we may have information that somebody had ibuprofen, or that they may have had Advil, or that they had an oral tablet of some specific dosage. And being able to represent all of these components together and make sense out of them requires utilizing this hierarchy. So thankfully, there's a lot of that we can leverage and that we can map source data into. But this gets really complicated with new concepts, new kind of drugs, new kinds of medications, and if there's multiple concepts. So think about like Motrin PM. Is that an NSAID or is it an antihistamine? It's both, but how do we really represent that within the data? So our team spends a lot of time thinking about how to be able to make those representations so that when it comes time to do the analysis, we're able to identify the correct data and be able to make generalizations at the right level. Another important component to this is evaluating the data quality. So as we put these into a common data model, we've done the standardization into some vocabulary hierarchy. We look at several different components of data quality. We verify and validate the conformance, completeness, and plausibility of the data sources, both of the source data and of how we mapped and harmonized that data. So we make sure that we didn't introduce any errors. So some of the questions that we ask is, does the data comply with the standards? What are the frequencies of key data elements? Do we always have date of birth? Do we always have gender? Things along those lines. But what is the plausibility? Are the data values believable and reasonable? Things like, was anybody discharged before they were born? That might be a problem. It's a little facetious to say that, but it is actually something that can occur where sometimes we just have typo in the data entry. But other times it's more systematic. We need to go back and look at the data pipeline and understand where in the transformation process did this error occur? And is this something that we can correct, or is this systematic and something else that we need to go further upstream and be able to understand why that is an issue? So evaluating these data quality are able to give us a good idea of what is the utility of the data and what kind of reliance can we have when it's actually generating evidence. So just to kind of give an idea of what we do once we have all that data integrated, we take all these harmonized data sources, we do our data quality evaluation, and then we work with researchers, quantitative medicine specialists to generate analytical subsets. We identify patients with a computable phenotype. This is something that Mike Padula talked about yesterday, that when we're talking about definitions around terminology, we don't want to just look at a single code. We want to look at what really is the indication of this disease or of this diagnosis that we're talking about. And so understanding that we can subset the relevant data to create derived variables is an important component of the process. And then we hand that off to statisticians to actually do the modeling and apply the mathematical interpretations of that. So just kind of wrap up here, data storage is absolutely secure. That's one thing that we always focus on through each one of those steps that I discussed. We're constantly looking to say, what are the possible vulnerabilities? How can we ensure that we have the top level security? How are we ensuring patient privacy and respecting the patient's right in all of this, each step throughout the process? We apply consistent terminology and vocabularies as we standardize data from multiple sources, transport to a common data model where we align them to a single format, we include hierarchical representation, and then we identify patients and subset relevant data to create derived variables for quantitative models. Thank you.
Thank you so much, Will. So that was really enlightening indeed, all of the work that you guys at the Data Collaboration Center do. And now I'm going to invite our colleagues from Quantitative Medicine, that is Jagdeep Wadishetty and Megan Kalla, to talk a little bit about what exactly is it that they do and taking the data from Data Collaboration Center and transforming that data into actual solutions. So first up, Jagdeep is going to talk about transforming real-world data into actionable knowledge for drug development. Jetty.
So I would like to start with introducing the concept of MIDD, or Model Informed Drug Development, which was briefly highlighted by Will Roddy in the previous presentation. The idea is to leverage data and quantitative methods to de-risk decision-making in drug development. The first formal definition for MIDD was published in a seminal paper in 2007. The definition focused on application of statistical model, now commonly known as pharmacometrics in drug development, knowledge management, and decision-making. Then later on in 2016, the definition was revised in another seminal paper. Here, the quantitative approaches were broadened to include newer quantitative methodologies that included AI, ML, and digital data analytics. So moving on, trying to understand what constitutes MIDD, fundamentally, it can be divided into input, modeling, and output. At a high level, we take the data and transform the data using modeling to generate knowledge. As John and Ralph described in their presentations, we must transform big real-world data into smart real-world data so that we can generate actionable real-world evidence through mathematically identifiable approaches. Now, with regards to real-world evidence, we should have a specific goal or context of use in mind and the regulatory application in mind. So for example, how can we optimize trial and study design? Providing a little bit more detail around the three components that I just described. The input is typically the patient level data. In our case, it would be the real world data. Then we apply appropriate modeling methodologies by leveraging different types of data. This could include demographic, biomarker information, in order to quantify the sources of variability and to understand the disease dynamics. The output gives us information regarding the way the trajectory of disease progression, and this all allows us to build something called a clinical trial simulator to run different clinical trial scenarios and inform clinical trial decision. Megan, in her presentation, will provide a little bit more detail around the underlying modeling framework for the CTS tool and how the CTS tool can be used for clinical trial design. Next, I would like to take a bit of time to provide what are the different impacts that MIDD has, different impacts we have had using MIDD in drug development. In tuberculosis, there's been a suite of quantitative tools that facilitated first new drug approval in about 50 years, laying foundation for 20 novel drugs that are being currently developed. In Alzheimer's disease, quantitative solutions such as clinical trial simulation or clinical trial enrichment tools are being developed to optimize the design for individuals, optimize the design of trials for individuals with dementia. In PKD, models combining biomarker dynamics and disease progression has helped the first approval of first ever treatment to slow disease progression and also facilitate development of new drugs. Next, looking at use of biomarkers with MIDD in kidney safety, a composite measure of biomarker was developed to inform drug selection. In PD, a different modality of data here, imaging biomarker such as dopamine transport imaging are being used for subject selection and trials in early motor stages of the disease. In type 1 diabetes, the biomarkers such as islet autoantibody are being used to optimize subject selection for prevention trials. These trials have resulted in first breakthrough designation of a drug to delay T1D onset. We are currently in the process of EMA qualification of islet autoantibodies as a model-based clinical trial enrichment platform for T1D diabetes, T1D prevention studies. So with that, I'll hand it over to Megan to provide a case study in Fragix Ataxia around the disease progression modeling.
Thank you, JD. So now I will be discussing a particular type of modeling that is included within MIDD called disease progression modeling. And this kind of modeling uses mathematical functions to quantify and describe disease changes over time. And within disease progression modeling, there are three main classes that range from purely data-driven or all the way to fully mechanistic, when the mathematical descriptions of the underlying physiology and the disease mechanism information is available. But regardless of the specific class, all of these models explain trends in longitudinal data with individual patient level data available. And at a very high level, this schematic shows an outline or a roadmap of the key steps that are involved in the development of a disease progression model, where the end goal here is to have a tool that can be used to inform future clinical trials, to be used to generate insights and actionable real-world evidence from real-world data. So first is the identification of the mathematical descriptions that can best describe the trajectories of the endpoints or biomarkers of interest in the disease. And then we examine the available variables in our data set that can be used to explain the variability that's present in the population, both at the severity at baseline as well as the rate of progression. And these can be genetic information or patient demographics, as well as other endpoints that are available in the data. And we can also account for additional design aspects like dropout effect or placebo effect if we can confirm that the data will support the inclusion of these additional model components. And then we can simulate a symptomatic drug effect and use the models to estimate the sample size that could be or would be required to detect a significant effect. And in a scenario where we have potentially several contending models, we could use the sample sizes as a quantitative metric to compare those competing models or endpoints in the disease. And so to put some of this into context, I'm going to relate it to a case study in Fragix Ataxia, which is a rare genetic neurodegenerative disease. And the endpoint of interest here in Fragix Ataxia is the modified Fragix Ataxia rating scale, or mFARS. And that's shown here in the schematic on the y-axis. And this mFARS scale is an assessment of the neuromuscular function, and it ranges from 0 to 93, where 0 is no disability and 93 is the maximum impairment. And included in this scatter plot are the longitudinal mFARS scores for about 1,000 subjects. And when we talk about the sources of variability within these populations, we can consider the variability at baseline, which is when a patient would enter into a trial, as well as anywhere along the disease severity spectrum. And then we can also consider and investigate the variability with respect to the progression of the disease, or the rate of change as the disease progresses. And in Fragix Ataxia, the progression rate is correlated with the age of disease onset. And this can be visualized in this schematic by stratifying this scatter plot by the age of onset. So you can see the bins as shown in the legend at the top, ranging from the different bins increasing with increasing age. So this can be used to identify that later age of onset groups have a mild or more linear increase in the disease progression over time. In general, a faster progression in mFARS would correlate with a earlier onset of the disease. So in the case of Fragix Ataxia, the age of onset could be used to incorporate or account for some of the variability that is observed in the rate of the disease progression. And the math behind these models, the math can be thought of as the backbone of what enables these simulations to be run from the models in order to answer and to test several different scenarios to address certain clinical trial design questions. And I'm not going to go into detail on these different types of models or the specifics of the equations, but here are some examples that were chosen for this scenario in Fragix Ataxia. And when we test these models, we can use the various metrics such as the objective function value, or the AIC, which is shown on the far right of the table. And those metrics can be used to quantify the suitability of these various models and how well they're able to capture the trends that are observed in the population. And then after selecting the mathematical structure for these models, the individual specific factors are probed, and this is used to identify sources of variability within the population. And as I alluded to during the age of onset discussion with mFARS, we can incorporate these various features that present with observable relationships in the data. And so this slide here is showing a particular method that in which we stratify the population by various features that are available in the data set. And what we look for here are features that deviate from the horizontal line of y = 0, and that's shown by the dashed line in each of these plots. So for instance, in the case of FA, the two features that I've highlighted with the red dashed boxes, these would be good candidates for features that we could explore, because the data is showing that these features are deviating from that horizontal line, so there may be a potential effect that these features have in the data. And this step is iterative, and we would repeat this throughout the model development to ensure that the model well describes all of the different sources of variability. And so in summary, what I hope I've covered today in this discussion of disease progression modeling is how they can be used to describe the trends that are present in longitudinal individual patient level data. And through my case study in Fragix Ataxia, I introduced a specific modeling approach called mixed effects models as a means to quantify the uncertainty and variability in disease progression. And lastly, I'll conclude with a larger picture here of how these models become useful and what we really want to emphasize. And these models provide the foundation to support clinical trial design, and they're what can help us to turn real-world data into actionable real-world evidence. And in doing so, they can help us with sample size calculations, the determination of trial durations, measurement timing, and other design and key analysis questions that can be addressed through clinical trial simulations and ultimately generating insightful and actionable solutions. Thank you very much for your time, and we can address some questions at the following panel discussion.
Right. Thank you so much, Megan. Thank you, JD, and thank you, Will, for your excellent presentation. I think, what you talked about should hopefully be good to explain to people exactly what happens to the data once it gets there. What does Data Collaboration Center do, and what does quantitative medicine do to actually generate those drug development tools that we've been talking about since the day this meeting started. I'm gonna jump directly into the panel session today. We have an eclectic collection of panelists, experts in their respective fields who have agreed to be in our panel. So those panelists are Dr. Satoshi Kusoda, Mike Pedula, Laura Fabry, Andrew Brammer, Colin Hovinge, and Shirley Porkis. But before I actually get into this, I wanted to take at least a few minutes to invite, because as I said before, the impetus for us doing this real world data session today was the real world FDA-funded real world data project that Dr. John Concato had mentioned in his speech today. So just for the benefit of the audience, what exactly is it that we were talking about when we were saying about the neonatal INC's real world data project? I wanted to invite the two co-principal investigators of this project, that is Dr. Klaus Romero, who I've already introduced to you today, and then another one was Dr. Jonathan Lewis, who is also present in the panel today. He's the professor of pediatrics and vice chair of pediatrics and chief of newborn medicine at Tufts Children's Hospital. So those two people are the co-principal investigators of the study, and I wanted to invite them on stage to provide a very brief overview of what exactly is that project that we keep talking about, and what exactly is it that we want to accomplish in that project. So Klaus and John, if you are here and if you can hear me, I would certainly welcome you to start this panel discussion by providing people an overview of our project.
Happy to start.
Do you want to go first?
Yeah, sure. Yeah, I can just be quick because I also want to hear from the panelists. But The notion is this: we at CPATH had developed a tremendous amount of experience and expertise, and we have achieved tangible impact that JD showed with transforming drug development in tuberculosis, PKD, Parkinson's, Alzheimer's, type 1 diabetes. Inc. was there for that opportunity to be taken, and with the FDA's investment and drive towards really having an actionable look at the tangible applications of real-world evidence generated from real-world data. The notion is to take what we've learned and apply it to Inc., but also explore specifically the real-world evidence angle and make it actionable, as John Concata was presenting in his talk. So the aspect is make sure that we use this opportunity to turn big real-world data into smart real-world data through the work that Will described, and then take that smart real-world data and turn it into real-world evidence in the form of those actionable solutions that JD and Megan described, in a way that is very much tailored towards tangible regulatory application to accelerate neonatal drug development, specifically to case studies, bronchopulmonary dysplasia, and lab values in the NICU. Again, with a focus on how to accelerate drug development and turn those solutions from the real world data. And in the process of doing that, build a data and analytics platform around it. A component of this has to do with diagnosing what the data can actually support, which was another thing that John Concata insisted in his presentation. So this is, like I said in the beginning, the opportunity for us to really provide the neonatal community with a series of very actionable tools, but also accrue a ton of lessons that will be actionable for the neonatal community, but importantly for everybody else. And this is the opportunity that I really love, where the neonatal field will be teaching everybody else how this is done. So I'll stop with that. John, take it away.
Thanks very much, Klaus. First, I want to start by thanking Deb DeSenza because I agree with Kanwaljit. Her words were poignant and reminds us why we're here. We really need to help these babies, and all of you are contributing in a positive way, but we need to do more and we need to do better. We need to develop data sets that will allow us to develop predictive models to really help Deb and other parents know which children are at risk and which children are at less risk, and then to use our limited resources to helping those children achieve the best outcomes that they possibly have. And I spent most of my early career lamenting about regulatory roadblocks that were put up at FDA or EMA or other regulatory agencies that really prevented us from developing these kinds of new products to help these babies. And yet now we hear from our regulators, and I really appreciate John and Ralph's comments, they want to help us. And they've given us a unique opportunity to generate this large data set that we can use with the spectacular data science opportunities and abilities of Critical Path to answer so many of the questions that people like myself and many of you in the audience have had through your entire careers. But one thing I've learned, it's hard. It's really hard. People are really interested in working with us. They want to get us those data sources. They appreciate that this is our one opportunity to make a huge difference and impact in these babies. We can't possibly do all the randomized controlled clinical trials we need to do. This is a great opportunity for us in neonatology, one that I haven't had in my 35 years as a division chief. So we have to work with the companies. We have to work with the academic investigators. Dr. Brammer is on the panel. He has been a tremendous ally from NIH, and we've got the first sets of data from the NIH DASH. It's one of the first times NIH and NICHD in particular has shared data with these types of projects. And you really need to get people like Drew who will sit down, roll up their sleeves and say, there must be a way that we can do this. we have to do this with other companies. We've had people say, we're really interested in doing that. But then as we go through the pipeline, the legal blocks, the financial blocks, the really people wanting to control in a very strenuous way how the data are used, we understand all that. But it's those kinds of roadblocks that we really need to eliminate. So I'm really excited about the opportunities that we're given. I am not backing down. I will not give up on this. I think we really have a unique opportunity, and we need everybody on this call to work with us, get those data to us so that we can really make a significant difference. Thanks very much.
Thank you so much, John. Thank you, John. Yeah, exactly. That is exactly the purpose of having you present the background of this project is, as Klaus said, this project focuses on 2 aims, actually among a couple more, but I'm only focusing on the 2 clinical aims. One is we will, JD and Megan and others in quantitative medicine, along with will collect a large amount of electronic health record, registry, and clinical trial data to create a disease progression model and clinical trial simulators for bronchopulmonary dysplasia, which the neonatologists among the audience would know how important and how crucial that disease is. It defies treatment. It's one of the only ones that's been increasing in prevalence. And then the second one we're focusing on is creation of generalizable, actionable lab value reference ranges in neonatology, which, I'm pretty sure you would also know that this is something that is very deficient currently. So what we have done in the panelists, in our collection of panelists are actually most of the people in the panelists are also collaborators for us in our real world data project. We are working with each and every one of them and basically having them contribute data to this project. And I'm going to invite each of them to actually present their perspective and what exactly is it that they are doing for this project. And again, thank you so much to all of them in advance. First up, I'm going to invite Dr. Satoshi Kusuda, who is a neonatologist at Kyorin University and the head of Japanese Neonatal Research Network. And he's going to talk about what their his perspective and what exactly their contribution to this project is going to be about. So Dr. Kusuda, I'm going to put your slides on. And he's also a professor of pediatrics at Kyorin University in Japan. His presentation is on the real world data project in Japan for the CPAP IMC project.
Thank you, Kamarajit, for your kind introduction. I'm Satoshi Kusuda, an neonatologist working at Kyorin University in Tokyo. I'm so pleased for giving me this opportunity to make a presentation today. Today, I'd like to briefly explain what kind of contribution Japan can make to this real-world data project. This time, 5 hospitals with large NICUs will participate in this project and provide data. At these 5 hospitals, all laboratory test values from infant during stay in NICU are stored in electronic medical records. And procedures and medications are also recorded in DPC system, which is mainly used for reimbursement. Therefore, all necessary data for the real-world data project can be extracted from EMR. Furthermore, if an infant is less than 32 weeks of gestation, this infant is also registered in our neonatal network database. We can link the extracted data from EMR with this existing network database. The database includes the outcome at 1.5 and 3 years of age. So we can create a data set that contains both clinical data during NICU hospitalization and also information about after NICU discharge. Now we cleared the IRB process, so we plan to carry out the data extraction at each facility. This slide shows the collaborators involved in this project. I would like to thank those collaborators. So we would like to contribute this project from Japan as much as possible. Thank you for your attention.
Thank you so much, Dr. Kusuda. We greatly appreciate not only your contribution today to this project, but also your willingness and dedication to be actually present as a panelist in this for this meeting, right? Understanding that it's close to midnight where you are right now. So, double gratitude and thanks for being here. And, yeah, we also look forward to working with you on a mutually collaborative basis for a project that is going to be ending up as a win-win for everybody involved. And I'm going to continue on to the panel discussion further. Next up, I'm going to invite Dr. Mike Padula, who's the Medical Director of Informatics and the Professor of Pediatrics within Division of Neonatology at Children's Hospital of Philadelphia. And Dr. Padula is going to talk about forging a path forward for neonatal real-world data. Mike, so you're on.
Thanks, Kamaljeet. Yeah, I just wanted to briefly touch on some of the sort of conceptual changes as we think about how data has been collected previously to how it might change moving forward. Really taking the idea that, in trials we might have looked at a set of criteria and then assigned a binary diagnosis, was, next stage 2, based on Bell's 2A, based on Bell's criteria present or not, to a more granular approach to data collection where we, dependent on electronic extraction, where we're really looking for confidence that the diagnosis was assigned, accurately, and we're therefore leaning on evidence, leaning on criteria that the disease or condition is present, and in doing that, we're hopefully increasing our precision and quality of the data. And there are other implications then to how these data may be used. So I touched a little on this briefly yesterday, but the challenge then becomes, as we consider the conditions of interest, it really takes subject matter expertise to know how, what are the critical features that differentiate similar concepts, that, and how do you lean on capturing, those supporting criteria discreetly, so you can, differentiate when one is present versus another, and you can have confidence in the diagnoses or conditions that you're assigning are accurate. The other, real value in having this sort of granular approach is it really allows for more flexibility for staging and severity based on existing classifications, or even creating novel classifications for understanding a disease. As Megan pointed out, this disease progression modeling would really benefit from this more granular data collection. Additionally, you can sort of understand how prior definitions might compare to existing definitions, or a more rigorous definition might compare to a surveillance definition. So that's really another benefit or flexibility. And as I mentioned yesterday, it also allows for definitions to evolve. You can understand how a single, sometimes more challenging data point that's available in some places versus another, whether, how much that's contributing to the diagnosis. So as Will Roddy touched on really nicely, a lot of these sort of granular details that I'm mentioning, there's work to be done. This is sort of my call to action, if you will, to really understand what those important differentiating concepts are, these details of interest, and ensuring that there's a representation of these concepts among the reference terminologies like SNOMED or LOINC, so that these reference terminologies really support data aggregation and retrieval, and there's power in that that I won't go into. But those codes are the same whether it's in the U.S. or Japan with Dr. Kasuda or wherever. They're agnostic to any specific site. And the way this can be leveraged is just as in the details that Will covered, or I also just wanted to briefly mention an important evolving standard known as Fast Healthcare Interoperability Resources, FHIR, in which you get a lot of the data that exists within an EHR are sort of queued up on a server that can be queried, and that really enables a lot of flexibility for the secondary use of these data. And the richness of what data will be available really is dependent on how well we contribute and flesh out these other reference terminologies. So I will pause there, and I look forward to discussing more.
Thank you so much, Mike. Just going right into the next panelist, given the amount of time that we have left, I'm going to invite Dr. Laura Fabry, who is the head of clinical neonatology at KSD Pharmaceuticals. Just like Dr. Kasuda and Dr. Padula, KSD Pharmaceuticals is also collaborating with us on our real-world data project, and I'll invite her to provide her perspective.
Thank you all. And why I am here today as a pharma representative because, as said, I wanted to witness the commitment of KSD in this field. I have been working in neonatology in this company, which has the corporate in Parma, Italy, so far away from you. But neonatology was always important for us, and I have been working for 15 years when the clinical development of new product was very standard and based on randomized clinical trials, as it was for our surfactant. But now the attention of the potential use of real-world evidence to accelerate the development, as it is, for example, of a new product we are developing, a new form of buprenorphine, could be useful. So certainly from the time a few years ago when I attended expert panel discussion organized by our chairman today, Nina Modi, in Europe, something has improved in KSD, and now we have a specific area on real-world evidence, and we also wrote a vision for real-world evidence in next years. Let me read it. It is this one. By 2025, we will be recognized as a trusted partner for our stakeholders, ensuring cases reliability in generating real-world evidence to advance disease knowledge, patient care, and value proposition of corporate brands. Real-world evidence as one integrated voice will be part of KSD's innovative culture and expertise, and will be embedded in our portfolio strategy, product development, and life cycle management. This is to say, friends, that we are moving from a current use of real-world data and real-world evidence for phase 4 of development at affiliates level to inform clinical practice on our in-market product and establish a relationship, for example, with healthcare professionals, or for post-approval safety studies requested by regulatory authorities, to a near future where we want to use this data for our product development, to also evaluate the potential reimbursement, and why not in selected cases also for regulatory decision. I was happy to hear the words by our speakers from EMA, from FDA, the importance to start the discussion with them soon for these possibilities. And this is finally, just 2 minutes more, to say to you this is the main reason why we are collaborating with pleasure to this CPATH INC program on real-world evidence to generate knowledge in bronchopulmonary dysplasia and lab values for neonates. I have to say with satisfaction that for the first time, cases signed a data transfer agreement to share data from our cases-sponsored surfactant studies that gave useful data and to be grouped with academic data, other companies' data for a common multidisciplinary database, font certainly of future information with different applications just mentioned. So this is the reason why I am also here with you. Thank you again on behalf of Casey.
Thank you so much, Laura. We're so happy that you could join us today, and obviously we're very pleased on our very productive collaboration with KSE. The one that we just worked on is only the first one of many, which I hope we will continue working with you on many such more collaborations in the near future. Thank you so much again. Next, I'm going to invite Dr. Andrew Brammer, who's the Chief of Pediatric Growth and Nutrition Branch at NICHD. As John mentioned, and as Klaus also mentioned, this is really the very first time CPATH has ever collaborated with NICHD on any of the data sharing projects. So we are doubly happy and proud that and honored that Dr. Brammer could be here and be a part of the panel and provide us with his feedback with the NICHD hat on. So Dr. Brammer, I would ask you to provide a few comments as well.
Wonderful. Well, Kanwaljit, I thank you and John, and I just want to I wanted to just say thank you. It's the first, but definitely not the last. So this is
absolutely
the beginning, but I want to start going back to and just thanking Deb. I mean, for a powerful presentation this morning. All of us here want to do the right thing, and this is people that we're talking about, and there's nothing more important than the human capital that is here on the screen here as part of this group, and as clinicians that we see day in and day out. I have the absolute privilege of working at the NICHD, where we do focus on pregnancy and child health and human development and the entire lifespan. And it's such an honor to work in this space, particularly with neonatologists who are devoted to doing the right thing and taking care of families, not just physical health and ailments, but also mental health, and emotional health, and holistic health, if you will. And so when John and I had our initial discussions, it was one of those aha moments. That's by golly, we have all these different activities in this space, and everyone wants to work together. Are there ways that we can be creative and make it happen? And the answer is yes. And that's nothing special to me. It's a matter of having the conversations, sitting down together and figuring it out. And then so I want to thank everyone that's been a part of this group. Again, I have a tremendous honor and privilege to be where I am, but that's only because I work with such amazing people, and seeing the fruition of hours and days and months of trials, years of trials, make its way to impacting clinical care and changing lives of the families that we take care of. There's nothing better than that. And so I could go on, but I want to thank everyone who's been a part of this process. I want to thank CPATH. I want to thank John for their dedication and devotion to making a difference in the lives of those that we care for in the hospital, and not just in the hospital. Going back to today's presentation, these are data, these are decisions, these are medical management opportunities that impact people's lives for the duration and impact families. And so with that, it's an honor to work with you. I hope this is the first of many such opportunities to leverage the resources and the data that's housed at the NIH and NICHD in particular, and move it in a way that's maybe wasn't accepted 10 years ago, but now it's a way that really can make a profound impact.
Thank you so much, Dr. Brammer, and I absolutely echo your thoughts. This is the first, but certainly not the last. Absolutely. Thank you. Thank you again. Next up is Colin Hoenge, who is the Senior Vice President of Clinical and Scientific Affairs at Iact for Children. And we are actually working with Iact for Children and utilizing their clinical site network to get a lot of EHR data. So thank you so much, Colin, for being a part of this panel today. And I would love to hear your thoughts as well.
Sure. Thank you, Konalji, for having me. I want to thank also the leadership of this grant as well as the FDA, because it's really, from my perspective, a culture change. I think it's one of the biggest, most ambitious projects that has the greatest potential for impacting children's lives. You know, one of our roles, we're a public-private partnership, also in part supported by the FDA, not only to foster a clinical site network, but also to provide advice and guidance to pharmaceutical companies. And where I struggle in those steps sometimes is really having a really good data set to be able to access, that I can get to information in a very strategic way. In many cases, the existing structures are fairly lacking, for lack of a better word. You know, I want to stress that our sites have been really excited about this. I think that they see this as a potential to give back. I also see that there's a lot of enthusiasm about academic pursuits to expand things. So there's a lot of corollary activity. In the heart of COVID, we had sites where our response rates for general feasibility surveys for clinical trials were fairly significantly small. About half sites respond and express sort of this battle cry for interest. So I think at the end, as this begins to synthesize information and to create structure, it has really market potential to change the landscape in neonatology. An invaluable resource. I commend you all for the activities, and I'm happy to be a participant in the effort.
Thank you so much, Colin. As you know, as you mentioned, and as pretty much everybody knows, that Iact for Children has access to a huge network of sites, and you know, your collaboration and partnership on this project is really, really critical and vital. So I'm thankful for you and for Iact for your collaboration on this project, and I'm sure we'll make excellent progress in the months and years into the future for this particular purpose, and not only for this purpose but for other projects as well. So thank you. Last but not the least, I'm going to ask Dr. Shirley Porkes, who's the chair of the Policy and Communications Group and Vice President-elect Faculty of Pharmaceutical Medicine and Director of Actors Consultancy in UK. So Shirley, would you provide your feedback as well as someone from UK? Because, you know, as you see, most of the individuals who we spoke either from are from the US, so would love to hear your perspective as well. Thank you.
Thank you very much, and thank you for the invitation and to share a few words on an industry perspective. I'm a pharmaceutical physician, so a medic. I've worked in industry and medicines development and policy for a number of years, and I'm about to become Vice President of the Faculty of Pharmaceutical Medicine, which is a professional body for doctors working in research and the development of new medicines. And our mission is to advance the science and practice of pharmaceutical medicine for the benefit of the public, which is, you know, what today's about. So it's great to be part of this discussion. And I think for us, real world data is exactly one of those areas where the science and practice of pharmaceutical medicine is evolving, and quickly, as we've already heard. And our members who work in companies, work with regulators, or work with research organisations, they're working with the issues that we've talked about today. And as FPM, we have held joint workshops earlier this year on data and pharmacoepidemiology, and data will be part of our symposium next month. And we need to think about how we turn that data into those insights to give us answers to those questions, becoming real world evidence, as speakers have already discussed today. But we have to do that whilst evolving our current view of real world data, you know, from data in health records and product registries and wearables to whatever comes next, which will be driven by the technology. but also in the context of policy decisions about how data should be used. And the industries, as we've heard, is keenly interested in those data sets. So ABPI, which is the UK Trade Association, published a report last year which talks about how health data is being used in developing new medicines, from understanding disease and defining unmet need through to proving the value of a medicine in the real world. And FPA, the European Trade Association, has highlighted the need for a real world data and evidence regulatory framework, with principles for data quality and interoperability and regulatory acceptance as part of their regulatory road to innovation. So my reflection is that medicines development needs 4 things from data. Firstly, the data needs to be high quality, as Nina said right at the beginning, with breadth, depth and scale. And it needs to be multimodal or linked data sets, which is I love Will's analogy with the plugs and the cable. Secondly, the data needs to be accessible and easy to use and cost effective to access for organisations of all sizes. Thirdly, expertise in data science and artificial intelligence. We need that data science to help us collect and analyse that data so we draw the robust conclusions. And finally, most importantly, at the heart of all of this has to be trust. And it's got to be trust by industry in the robustness of the data, trust by the regulator that enables the data to be used in licensing, trust by parents, carers, patients in how the data is being used, and trust by the public in those governance mechanisms, and particularly where there's a commercial aspect. And understanding what's needed to both develop but also maintain that trust as the field moves rapidly absolutely needs ongoing conversations. So conversation like today's panel is brilliant to say, okay, how do we actually bring those perspectives together? And really happy that the faculty is able to be part of today. Thank you.
Thank you, Dr. Porkis. Again, as you said, trust, that's the most important thing, and that ties back into the comment that was made by Ralph Bax. How do we avoid having these type of solutions end up in sort of a black box? Trust is something that'll certainly help in quite a lot way by avoiding those type of misconceptions. So thank you so much to the panelists. We have only about 3 minutes left into this part of the session. There is one question that has come in from the audience that I would like to ask from Dr. John Concato. That was from Jerry Bear, and that question was, are there commonalities to the products and programs that have incorporated real-world evidence for approval? For example, are the endpoints relatively standardized or objectively assessed?
Thanks, Jerry, for the question. I'll give a big picture answer and then try to hone in. I think it might be fair to say we're relatively early in the real-world evidence era. And I don't think it's going out on a ledge to say that commonalities will emerge and outcomes are a critical aspect. So I think we have to find that balance point where we're not too passive and saying, well, we don't know yet, but we're also not too prompt to rush off and use endpoints that aren't that turn out not to be what we need, not fit for purpose, as we say. So it's more than just saying it's a case-by-case example, but I think the field is advancing and this initiative can help. If I may, and I apologize, I have a hard stop at the top of the hour, but related to Jerry's question, I just want to say that one way to put it informally is drug outcome associations are agnostic to our conceptualizations of clinical trials or real world evidence. I mean, at the end of the day, I'm not putting too much on the epidemiologic underpinnings, but we have to focus. I think John Davis said it's hard. It is, but if you don't mind, one step at a time, we can have a mix of safe and risky ideas. I don't mean risky drugs, risky ideas. We shouldn't be too tentative, but we really have to do it one step at a time and do it right. I think at the methodologic level, that's why it might sound very unexciting to say, oh, standardized lab values. If we don't get that right now with this project, then every project that follows is vulnerable to a problem with the lab. So we don't want to be, again, too slow, too passive, but we also don't want to rush off and cut corners. So it sounds like a platitude, but so be it. Do it right at the basic epidemiologic level. And then part of that links back to Jerry's question about endpoints. I think this is multifaceted. We heard about model-informed drug development. Everything we could do intersects almost always with other aspects. So sorry to have to sign off, but this has been a pleasure to be here. Again, FDA is committed to doing what we can to participate in this process, but mainly we're looking forward to the great work of coming out of this project and of the large group that's been assembled. Thank you very much and have a great day.
Thank you so much, Dr. Concato. Thank you again, and thanks for joining in. Look forward to catching up with you again soon. Thanks.
Bye.
Okay, so we at least the panel discussion is pretty much over, but we wanted to take a few minutes to recognize a couple of certain collaborators of CPATH INC that we have been working with over the past 1 year for the real world data project that Klaus and John Davis had talked about. And INC is very, very proud and honored to start instituting a Data Pioneer Award series from this year. The purpose for this award series is basically to honor and recognize the contributors and collaborators who have been especially very active in data and helping us with our data sharing and data integrating activities. For the purposes of this award, I'm actually going to ask back our parent collaborator, Deb Dicenza. She's going to basically be honoring all of our data contributors and providing the awards to them. So Deb, you're on next. Just let me know when you want to progress to the next slide. I'm happy to do that.
Thank you. Oh, we have slides. Okay.
Yes, we do.
Wonderful. Well, I'm going to start with a quote. None of us, including me, ever do great things, but we can all do small things with great love. And together we can do something wonderful. Mother Teresa. I am doing this in memory of Patrick Bouvier Kennedy, who is the son of the late former President John F. Kennedy and his wife. And they lost their baby to what we now know as respiratory distress syndrome. So thank you. I am honored to be here to give you these awards. And the first person is Tom Miller. Dr. Tom Miller is the Vice President and Global Head, Pediatric Development for Bayer Pharmaceutical Division. He's also part of our Inc. leadership team and a wonderful member of that, by the way. He's been fantastic. Bayer and Dr. Miller, they have shared their clinical trial data from Bayer. Thank you, Tom.
Thank you, Deb, for that very kind introduction. I'm going to take Susie McCune's lead from yesterday and give an actual live visual of the award that I'm very honored to accept on Bayer's behalf and my many colleagues there that have made this possible. I'll go back to a point that John Davis made a few moments ago as we were introducing the session, in that this is a brave new world for all of us, and including those of us at companies. It was not easy to navigate through the process and the complexities of the company. He was spot on, but it is possible. So I know that I have a great industry partner in crime coming up next on the hit parade that has been able to navigate through the complexities as well at Chiesi. But I would encourage all of the companies that are participating in the session today, if you need any help or assistance in trying to navigate through the network, any tips that we can be helpful with, we're more than happy to do so, because like most things in life, the most important things are worth the effort. So thank you, Deb. Thank you, INC colleagues that I'm privileged to serve on the leadership team with, and thank you, CPATH.
Wonderful.
Thank you so much, Tom. From all of us, on behalf of all of us at INC and CPATH, I would also like to thank you for your collaboration. For CPATH, certainly this has certainly been the first time ever that we have worked with Bayer. So it's a matter of great honor for all of us at CPATH and INC. Thank you again. Deb.
So our next person is Dr. Laura Fabry, Head of Clinical Neonatology and Global Clinical Development at Chiesi Pharmaceuticals. Laura has consolidated relationships with worldwide neonatologists because of reciprocal faith and friendship due to the long-lasting collaboration. She is a member of the International Neonatal Consortium, committed to organizational plans, processes, improvement, and global standardization in neonatology research. Dr. Fabry and Chiesi, they are committed to sharing clinical trial data, and I thank you. Dr. Fabry.
Exactly, Deb. Thank you all again. I have just received this award from from the other part of the ocean. So thank you. And it is my privilege today to receive it, and on behalf of Chiesi, I wanted to say that was unexpected and a wonderful acknowledgement, and it is like a new sign of your friendship, respect, and the wish to collaborate all together. I can say the same words of Thomas Miller on how complicated sometimes is in a company to receive the approvals for sharing data, but we won. And we are extremely happy to do that in collaboration, of course, with many of my colleagues. And let me give you also personal thanks by Dr. Paolo Chiesi, who is the father of neonatology in our company and who has first recognized 30 years ago the importance to collaborate with the academy. And together with the researcher from Karolinska Institute in Stockholm, identified the poractant alpha cursurf and the potential benefits of it for premature neonates with respiratory distress syndrome. So Chiesi is in INC and in these fields because we are really aware and want to continue with this collaboration with academics and also regulators. So thank you once more. And the parents, Deb, I forgot, sorry. We are more and more recognizing the importance of your voice in all our development. Thank you so much.
Thank you. Thank you.
Thank you so much again, Laura, and thanks, Deb. Laura, your collaboration with our collaboration with Chiesi is really very critical, and you guys have been very gracious and with sharing your clinical trial data with us. And as I said before, this is just the start, and certainly not the end, and we look forward to continuing this collaboration with you.
Wonderful. Thank you. Next is Dr. Andrew Brammer, Chief of Pediatric Growth and Nutrition Branch with the Eunice Kennedy Shriver National Institute of Health, Child Health and Human Development. That's a lot of words. He is also the acting chief of the Pregnancy and Perinatology Branch at NICHD and the program official for NICHD's Neonatal Research Network, Maternal Fetal Medicine Units Network, and Global Network for Women's and Children's Health Research. Dr. Brammer was Inc.'s liaison within NICHD and helped us gain access to high quality study data from the DASH database. That's a big deal, everybody. So if they can do it, you can do it. Dr. Brammer, thank you.
Deb, thank you. No, I love that Mother Teresa quote that you started with, and I also I'll also circle back on Patrick Bouvier Kennedy, because as you mentioned that he died at 39 hours of life, and that really was it was Stephen Livingston as an author that actually coined that as the loss that transformed JFK, and as you mentioned. The Institute was founded by President Kennedy and it was renamed after Eunice Kennedy Shriver years later. And so there's a history and legacy, and certainly our mission is to do the research and training to really push the field forward in any and all ways that we can. And so I don't have my award here because I brought it to the Institute to put on display, and because that's where it belongs. It's as we're seeing today, these awards don't go to individuals; they go to groups of people, that come together to make it happen, and that is investigators, that's administrators, that's academics, that's the private sector, that's government, non-governments. We're all in this together, and so and even though I'm being called out, I also want to, in full disclosure, it was the individuals and the investigators at the various at DASH, at the data coordinating centers, John and his group, CPATH, everyone came together. Yeah, I just helped convene, and so I don't want to take any credit that's not due, except for the pleasure of being here and saying that this is an endeavor that we want to continue, and I look forward to many more years of these discussions. So thanks, Dave.
It always begins with a conversation and a meeting. So thank you.
Thank you, Drew. So that particular collaboration that we talked about, I am very sure that's just one of the first, and I'm sure I'm going to be pestering you for many more of your similar data sharing requests in the upcoming weeks and months. Thank you so much again. It's such an honor for us to provide you this award, and thanks for joining in for the panel discussion today also.
And last but not least, Tufts Medical Center. Dr. Jonathan John Davis is the Vice Chair of Pediatrics and Chief of Newborn Medicine at the Floating Hospital for Children at Tufts Medical Center and Professor of Pediatrics at Tufts University School of Medicine. He is also the Chair of the Neonatal Advisory Committee in the Office of the Commissioner at FDA, the Director of the International Neonatal Consortium, FDA EMA, and a member of the Pediatric Policy Council representing the American Pediatric Society. He's also part of our INC's leadership team, co-principal investigator for the Real World Data Grant, and recently appointed as CPATH's Senior Advisor in Pediatrics. John, this is a real honor to be able to hand you this award, even though I can't do it physically, but it's an honor. Thank you so much for all you do for INC and for all the families.
Thanks so much, Deb, and here is the award that I accepted on behalf of Tufts and our Clinical Translational Science Institute. So all the leadership wants to thank you for this wonderful award for CPATH. It means a great deal to us. We really all worked very hard to cut through as much red tape to get you our clinical trials data as soon as we could. And we're working with CPATH scientists on de-identifying our electronic health record data and hope to get 7,000 to 10,000 infants to you as soon as we possibly can. This has been a labor of love for me because I recognize the unique opportunity we've been given. We've had some wonderful collaborators. And again, going back to President Kennedy, when he talked about going to the moon, we're using that theme. We're not doing it because it's easy, but we're doing it because it's hard. It has been hard, and yet we've made tremendous progress with our colleagues in Japan. Nina Modi in the UK has been wonderful working with us. I talked about Drew previously. The companies have been stepping up and getting us those data. So we're really hoping this will make a difference. We recognize in working with our regulators, it's not going to replace our randomized controlled trials. We have to continue to do those to get new drugs and existing drugs together. But it will help if we can look at 50,000 babies who've been exposed to a drug that's off patent and being used routinely in the NICU. Maybe we can generate some data on safety and efficacy that will help the regulators say, yes, you should be using it. Or if the drug is not safe, stop using it. There's been many drugs that we've used in neonatology for years, only for someone to look at the data finally and say, it actually didn't help and it was harmful, and we've stopped using it. So I do think that this will play an important role. So I do want to thank you and thank all of CPATH and our collaborators for assisting with this project.
Thank you so
much. Thank you again. Yes, I echo your comments, John. Thank you so much for everything that you've done. We, are all grateful to all of our Data Pioneer awardees for their exceptional efforts in promoting CPATH and INC's data integration effort. Thank you so much again from the bottom of my heart. And then next, I'm going to invite the session chairs, Klaus Romero and Nina Modi, back up again to provide closing remarks.
I'll go first, and then I'll give Nina the honor to close things. I'm really excited about this, and I'm not just saying this. Inc. and thanks to you guys, and these awards are very meaningful because Inc. has broken speed records for data acquisition and integration. No other effort at CPATH has shared data and integrated data at the speed that Inc. has done it. Of course, we wish things could move even faster, but you guys are moving at breakneck speed. So thank you. The patients and their families are going to benefit from this tremendously. There were some questions about how to join us. You can contact me. You can contact Kanwajit. You can contact Adriana Burns or Sarah Spieth or Laura Butte, any one of us at CPATH, and we'll take it from there. We're at time, but I'll stop with that. Thank you so much. Nina, please close the meeting.
Thank you very much. What can I say? It's been a fascinating meeting. I think the phrase to use is the future's bright. But I'd also like to say that what's great is the spirit of collaboration. It's wonderful to have colleagues, not just from the United States, but from all around the world collaborating on this. Satoshi from Japan, Laura from Italy, colleagues from all over Europe. It's been great. The world is a bit of a dodgy place at the moment. It is somewhat fragmented. So gatherings like this and collaborations like this, which are really going to improve the lives of our most vulnerable patients, are really heartwarming. So what a pleasure to be part of this. Congratulations, everyone, on a lovely afternoon, evening for us over here, and look forward to working together with all of you. Many thanks, everybody, and well done, Kanwaljit, for fielding these 2 days so successfully.
Thank you so much. Thanks, Nina. Thanks, Klaus. Thanks, everybody in the panel session, and thank you for our Data Pioneer Awardees. Thank you so much again, and thank you to the audience for actually sticking with us over the past 2 days. I hope the meetings were enlightening, interesting, and helpful to you. Thank you for coming in, and I look forward to seeing you again next year, hopefully in person, depending upon how things are. But again, yes, thank you from everybody at CPATH and INC side for attending the INC annual meeting this year. I look forward to connecting with you again next year. And if there are any questions at all, please feel free to be in touch.
Bye-bye, everyone.
Bye.
Thanks,
Thank
everybody.
you. Thanks.
Bye.
Stay safe, everyone. Take care.
Cheers.
Bye-bye.
Bye-bye.
Bye.
Bye-bye. Bye-bye.